Assalamu alaikum, students. In this lecture, we will learn about prokaryotic and eukaryotic cells. Prokaryotic cells. Let's discuss some basic points regarding prokaryotic cells. Prokaryotic cells are simpler cells. They are the most primitive cells ever present on the earth. So, cell wall is made of peptidoglycan and murines. Have not nucleus, but its genetic material is in the form of the chromosomes located in the cytoplasm. It is thus in the form of a bunch of chromosomes present in the cytoplasm. They are mostly unicellular. They are just 1 to 5 micrometer in diameter. They are too much small. Don't have any membranous bounded organelles. They have 70S ribosomes. Other structures like flagella, cilia, pili, fimbri, and plasmids are present in some eukaryotes. They are not present in all the prokaryotes, but they are present. Sorry, it should be right here. Prokaryotes. They are not present in all the prokaryotes, but in some prokaryotes. Archaea and bacteria are the two different types of the prokaryotes. They have the very minute differences between them, right? Division. They divide by the binary fission. Structural features of the prokaryotic cells. Capsule. Capsule is the present outside of the cell wall. It is involved in the moisture retention and helps to attach the bacteria to surfaces and the nutrients. They are involved to attach the one bacteria to the other and to attain the nutrients from outside environment. Cell wall is the outermost layer and gives shape to the cell. Jelly-like material made of the salts, enzymes, and the cell organelles are present in it. Cell membrane. It is a semi-permeable membrane around the cytoplasm. First of all, there is a cell capsule in some bacteria, but not in all. But in all bacteria, cell wall is present, then cell membrane, then pili. Pili are the hair-like structures that attach to the other bacteria. They help the one bacteria to attach with the other one. Flagella are whip-like structures and help in the movement of the cell. They are involved in the movement of the cell so that the cell can move from here to from one place to another place and can locate its food. And ribosomes are involved in the protein synthesis. Remember that ribosomes of the prokaryotic cell is 70S. Plasmids are non-chromosomal DNA involved in the reproduction. Because they do not have any nucleus, they have only a bunch of the chromosomes present in the cytoplasm. It can be said to be the nucleate region. And in addition to this, plasmids are also present, which are the non-chromosomal DNA, and they sometimes play a role in the reproduction, not always. How do the reproduction occurs in prokaryotes? It can be through asexual reproduction or through sexual reproduction. Asexual reproduction through binary fission, in which DNA of the bacterial cell duplicates and attaches itself to the cell membrane, and cell membrane invades its side and divides the parent cell into the two daughter cells. It is a simple. Sexual reproduction. Sexual reproduction takes place in three ways, including conjugation, transformation, and transduction. Conjugation means the genes are transformed between the two cells through a tube-like structure, pilus. A tube-like structure is formed between the two bacteria, and the genes are passed from the one bacteria to the another and from the second bacteria to the first one. Transformation is bacterial cell takes in the DNA from its environment and incorporate in its own DNA. For example, we have to make a lot of copies of a particular gene. We will put it into a particular test tube, and then in the same test tube we will add the bacteria, and then bacteria will take it that particular gene and then will incorporate it in its own DNA, and so we can say that the bacteria is transformed. In transduction, genetic material is entered in bacterial cell through virus, for example bacteriophage. In this process, an external source, especially a virus, is used so that the genetic material can be entered inside the bacterial cell. This is the structure of a typical prokaryotic cell. These are the structures which are always present in the prokaryotic cell. and they are sometimes present in that particular cell. So cell wall, cell surface membrane, and cytoplasm, circular DNA, and ribosomes, they are always present. But flagella, capsule, infolding of cell membranes, plasmic reticulum are present in sometimes, but not always they are present. Now come towards eukaryotic cell. Eukaryotic cells are somehow complex than that of the prokaryotic cells. So let's start. Cells which contain membrane bounded structures and other membranous organelles are eukaryotic cells. For example, protozoa, fungi, plants, and animals, they all are eukaryotic because they all have membrane bounded nucleus and the other membranous organelles. So reproduction occurs through mitosis. Their reproduction occurs through mitosis, in which the spindle fibers and centrioles are involved, and they are involved in the separation or segregation of the chromosomes, and results in, and then cytokinesis takes place and results in the formation of the two daughter cells from the one parent cell. So we will discuss it in the next lectures. Structural analysis of the eukaryotic cell is given in the next slides. Whatever the organelles are present in the eukaryotic cell, we will discuss further them in the next slides. Cell wall. Cell wall is only present in the plant cells. It is made up of the cellulose, hemicellulose, and pectin. It provides structure and support to the plant cells. Its function is just to provide support and a particular structure to the plant cell, nothing else. It protects the plant cells from the external injuries, as it supports its structure so that it will prevent it from the external environment as well. Cell membrane is approximately 7 nanometer in diameter. It is also known as plasma membrane, and it is semi-permeable, that it allows only a few materials to pass through it. All the materials cannot pass through it, right? So it is the outermost layer in the animal cells, and it is present next to the cell wall in the plant cells, because in the plant cells, cell wall is the outermost layer. It is porous and allow only a few molecules to pass through it, as I have already told you that it is selectively permeable, so few molecules will pass through it. Made up of the lipid bilayers and proteins. According to the fluid mosaic model, plasma membranes are made up of the lipid and the proteins, and the lipid bilayers in which protein molecules are embedded. Microvilli. Microvilli are finger-like extensions of the cell membrane to enhance the surface area of the cell membrane. Microvilli are basically the extensions of the cell membrane. For example, this is the cell membrane and they are extensions. They will just enhance the surface area so that the materials can be absorbed into that particular cell, or it increases the surface area so that the intake of the materials can be done easily. It is involved in the absorption of the materials in the gut and reabsorption of the substances in the proximal tubule of the kidney. Whenever in the kidney the filtrate is present, glomerular filtrate is present, and when it reaches to the proximal tubule, then some of the substances are reabsorbed. So here the microvilli are present in the cells, and they are involved in the reabsorption of substances. They are mostly present on the epithelial cells, not in all cells. Cytoplasm, they are present in the both animal and the plant cell. It is the base of the cell, in which the cell organelles are present and perform their different functions. It is a jelly-like material present between the cell membrane and the nucleus. It is made up of water, organic and the inorganic molecules. All cell organelles are embedded in the cytoplasm. And they perform their different chemical and the metabolic reactions in the cytoplasm. Plasmodesmata are strand-like structures through which the plant cells join with each other without invading in the cell walls. They are just the structures which join one cell to the other cell, so that they can pass on the different materials from one cell to the other cell. For example, salt, sucrose, amino acid, water molecules, etc. And this typical kind of the movement of the molecules from one cell to the another cell through the plasmodesmata is symplastic pathway. Movement of the material through symplastic pathway. And the plasmodesmata do not only involved in the transfer of the materials from the one cell to the other, but they are also involved in the cell signaling process. For example, a particular signal has to be moved from the one cell to the another. It can be done through plasmodesmata. Nucleus. Nucleus is 7 micron meter in diameter as viewed through the electron microscope. So it is a double membranous organelle found in all the eukaryotic cells. It is surrounded by a porous nuclear membrane. It contains DNA responsible for all the genetic activities of the cells. It contains chromosomes and the nucleolus. Nucleus is responsible for inheritance because it has genes, and the genes are that particular material which has the information for all the inheritance. So we can say that nucleus is responsible for inheritance. Nucleolus 2.5 micron meter in diameter. This is most important. This can come in the MCQs as well. So it plays in synthesis of the RNA and proteins. This is the structure of a typical nucleus. As we can see here, this is the nucleus envelope. These are the chromatins. This is nucleolus. These are nuclear pores, and this is the nucleus structure. And this is the structure of the nucleus. Around the nucleus, cisternae are present. That means this is a rough endoplasm which is present around the nucleus. Endoplasmic reticulum. A network of the fluid-filled membranous canals. They are just like a canals, a network of the canals which is present around the nucleus. It is responsible for the transport of the materials throughout the cell. If a particular material has to be transported from the one place to the another, then endoplasmic reticulum will perform that particular function. It is of two types, rough endoplasmic reticulum and the smooth endoplasmic reticulum. Rough endoplasmic reticulum is made up of cisternae, tubules and vesicles. They are found throughout the cells and involved in the protein synthesis because ribosomes are attached with the rough endoplasmic reticulum. That's why we can say it is involved in the protein synthesis. Smooth endoplasmic reticulum, no ribosomes are attached with it, so it is not involved in the protein synthesis. It is involved in the storage of substances and the production of the lipids and steroids, and it also detoxifies the cell. That is, it removes the waste materials from the cell as well. This is the structure of a typical endoplasmic reticulum. As we can see here, this is the smooth endoplasmic reticulum because it do not has any ribosomes here, and it is rough endoplasmic reticulum. The granules which can see here are the ribosomes, and we can see there is a nucleus around which the endoplasmic reticulum is present. Mitochondria is 1 micron meter in diameter, double membranous organelle found in all the eukaryotic cells. So mitochondria are involved in the production of the ATP, as the process of the aerobic respiration is involved in the production of the ATP. So this particular function is occurred to be in the mitochondria. Among the double membranes, one membrane form a boundary wall to separate it from the cytoplasm, and the other membrane form the infoldings inside towards the inner side of the mitochondria called cristae. Mitochondria is either round or oval in the shape, as we have seen it in the above pictures. It consists of its own DNA, RNA and the ribosomes for the protein synthesis, so that it can synthesize its own proteins itself. It is involved in the aerobic respiration, produces ATP and helps in the transformation of molecules. So it is the powerhouse of the cell because it is involved in the production of ATP. They are involved in the aerobic respiration and produce ATP, which is used by the cells for performing various cellular activities. Obviously, when energy will produce in the cell, then the cell will use this particular energy to perform its different functions. This is the structure of a typical mitochondria. As we can see here, this is the outer membrane, this is the inner membrane, which forms the enfoldings called the cristae, and some ribosomes are present here. The dot-like structures are actually the ribosomes, and the space, pink space, we can find out here is the matrix of the mitochondria. So these structures are here. You can show the DNA present in that particular mitochondria. Plastids. Plastids are basically involved in the process of photosynthesis, and they are of three types: chloroplast, chromoplast, and leucoplast. Each perform its own function. As we see through the electron microscope, we find out that chloroplast is 3 to 10 micron meter in diameter, and it is double membranous organelle present in mesophyll cells of leaves, made of stroma, thalakite membranes, and inner lamella. This is the structure of the chloroplast. Contains chlorophyll. It is a pigment which gives its green color, and it is involved in the photosynthesis. As we all know that chlorophyll is required for the process of photosynthesis, so the chloroplasts are involved in the process of photosynthesis in plants. It contains DNA, ribosomes, and enzymes for the protein and the carbohydrate synthesis. Now come towards chromoplasts. They are xanthophylls and carotenes, and they are responsible to give the different colors to the plants. When we see the different colors flowers in the plants, then we should know that this is due to the chromoplasts. They are responsible for the yellow, red, and orange colors in the plants. So now come towards leucoplasts. They are colorless and store nutrients. Their main function is to store nutrients, and we can further classify the leucoplasts depending upon the type of the nutrient which they store. So amyloplasts are those leucoplasts which store carbohydrates. Aleuroplasts are those that store proteins, and elioplasts store fats. This is the structure of a particular chloroplast. So we can find out that there is the outer membrane, and here is the inner membrane. Inner membrane has a particular lumen. The space inside the inner membrane is called the lumen. As we can see here, there are the thalakite membranes which are stacked together to form the grana or granum. Ribosomes are 25 nanometer in diameter. They are non-membranous organelle. They do not have any membrane around them. They are just as tiny round structures present inside the cells. The prokaryotic ribosomes are 70S and eukaryotic ribosomes are 80S. S is basically a Swerdberg's unit which is used to measure the ribosomes. Either present in the with the endoplasmic reticulum or freely moving in the cytoplasm. They can get attached with the rough endoplasmic reticulum, or they can be freely floating in the cytoplasm as well. They are composed of the ribosomal RNA and the ribosomal proteins, so that they can perform the protein synthesis for the cell. They are involved in protein synthesis as I discussed earlier. Golgi apparatus. It is also known as Golgi complex. It is a membrane bounded organelle made up of the cisternae. It is involved in the transporting, modifying, and packaging the proteins and the lipids. For example, a particular protein and the lipid is formed inside the cell. Then it has to be moved out of the cell. How it will move out of the cell? So the Golgi apparatus will pack these proteins and the lipids in the vesicles, and then these vesicles will be secreted out of the cell. They can be secreted in the same condition, or they can be modified in the glycoproteins or glycolipids as well. They are found in the cytoplasm. It is present in both animal and plant cells. It is involved in phosphorylation and glycosylation. For example, it can phosphorylate the proteins and the lipids, and also glycosylate that particular proteins and lipids. The main function of the Golgi apparatus is that it removes the first amino acid of the proteins to activate the proteins. For example, a particular protein is synthesized. It is not activated here. We need to activate the protein so that it can perform its function. So that's why we have to remove that first amino acid of the protein, that is the methionine, so that it can get active and perform its function as well. This is the structure of a particular Golgi apparatus. We can see that these are the this is the lumen, which is the empty space present in the Golgi apparatus. These are the cisternae, which are the foldings, and this is a secretory vesicle, which can contain either protein or the lipid, so that it can move out of the cell. So move towards the next slide. Microbodies. Membrane bounded vesicular organelles present in the both animals and the plant cells are known as microbodies. They are too much small that they are about 0.2 to 1.5 micron meter in size. They contain different enzymes and the proteins, and they are involved in the breakdown of fats, amino acids and alcohols, photorespiration in the plants, and the detoxification of peroxides. They include peroxisomes and glyoxisomes. They are present in some cells, not in all the cells, because the functions they perform are not needed in all the cells. They are required only in few cells. Peroxisomes involved in the catabolism of the D amino acids, polyamides and the bile acids. They catalyze the peroxides by peroxidases and catalases. Glyoxisomes are present in both plants and the fungi, and they convert the fatty acids to the carbohydrates so that they can be stored here. Lysosomes are basically the hydrolytic organelle. It contains hydrolytic enzymes, and whenever a particular old or a cell organelle or a particular old cell has to be removed out of the cell, and either they are removed out of the cell or they are degraded by the lysosomes. Cytoskeleton. Cytoskeleton is a network of the protein filaments that provides the support and structure to the cell. It is involved in the transport of the molecules, cell signaling and the cell division. It is made up of the fibers including microtubules, microfilaments and the intermediate filaments. Microtubules are basically the small tubes having a size of 25 nanometer. They are made up of two types of proteins, that is alpha tubulin and the beta tubulin. So these tubulins join to form the dimers, and dimers join to form the protofilaments, and 13 protofilaments are present in a particular microtubule. So how much complex is the structure of a particular microtubule, which itself is a very small, and they are involved in the cell division. Now come towards microfilaments. They are the protein fibers with a diameter of 3 to 6 nanometer, made up of the actin proteins, and they are involved in the cytokinesis and muscle contraction. Right? Intermediate filaments are 10 nanometer in diameter, and they provide the tensile strength to the cells. Cytoskeleton has some motor proteins including kinesin, dyneins and myosin. Kinesins pull organelles towards the cell membrane whenever they so that the organelles which have to be remain towards the close to the cell membrane, there the kinesin is required. Dyneins pull the cell organelles towards the nucleus. Myosin is involved in the muscle contraction, cytokinesis, exocytosis and endocytosis processes. So some important functions of the cytoskeleton are they provide the shape and the structure of the cell, and they are involved with the cell signaling and the formation of vacuoles and intracellular movements. Centrosomes and centrioles. They are involved in the process of division, that is the mitosis. So centrosomes are made up of the centrioles. Centrioles are 9 triplets of the microtubules. Their size is 500 nanometer, and they are involved in cell division. So we can see here, this is the structure of a typical microtubule, centriole and centrosome. Cilia are hair-like structures which are present on the cell membrane of the many cells, right? They are involved in the attachment of the one cell to the another cell, and they can be motile and non-motile. Motile means they are present in the respiratory tract of the humans, and they are involved in the removal of the nucleus, mucus. So non-motile cilia are involved in the cell signaling. Vacuoles mean empty space. Okay, they contain the enzyme starch, organic molecules, and the different substances like this. They can store the food substances, and they can also remove the waste materials as well. Plant cells have only one large vacuole which is present in the center of the cell, and the animal cells has many numerous vacuoles present in the cytoplasm. They are involved in the exocytosis and the endocytosis, and they have to maintain the target pressure of the cell. Now let's discuss some differences between prokaryotes and the eukaryotes. Prokaryotic are mostly unicellular, and eukaryotes can be unicellular and multicellular as well. And the prokaryotes are too small that they are just 0.2 to 2 micron meter in diameter, and eukaryotes are 10 to 100 micron meter. Cell wall is present in both. Nucleus is absent in prokaryotes because their nucleoid region is just a bunch of chromosomes, and it is present in the eukaryotes. And ribosomes of prokaryotes are 70s, and that of the eukaryotes is 80s. DNA arrangement is in the prokaryotes is circular, and that of the eukaryotes is linear. Mitochondria is absent in the prokaryotes because they do not have the double membranous organelles, and the eukaryotes it is present. Cytoplasm is present in both. Endoplasmic Reticulum is present in just in the eukaryotes. Plasmids are present both. Lysosomes are absent in the prokaryotes, and eukaryotes they are present. So cell division in the prokaryotes occur through either binary fission or through the process of conjugation, and in the eukaryotes it occurs through mitosis. So this is the last slide of this lecture, in which we will discuss about the viruses. Viruses are non-cellular infectious entities, and they are made up of the proteinaceous coat known as a capsid. Inside the capsid, a nucleic acid core is present, which can either be in the form of the DNA or the RNA strand. So they have a symmetrical shape. They have no characteristics of living cells. They replicate inside the host cells, and they use a host biosynthetic machinery to replicate themselves. So they do not have its own function to replicate. They just use the biosynthetic machinery of the host. So meet you in the next lecture. Till then, Allah Hafiz and stay tuned.